Good morning, friends. Today's I tell about you what is DNA replication. DNA replication is the process by which DNA makes a copy of itself during cell division. DNA is made of a double helix of two complementary strands during replication. These strands are separated. Each strand of the original DNA molecules then serve as a template for the production of its counterparts. DNA replication copying genetic information for transmission to the next generation occur in S phase of cell cycle process of DNA duplicating itself begins with the unwinding of the double helix to expose the bases in each strand of DNA. Each unpaired nucleotide will attract a complementary nucleotide from the medium, will form base pairing via hydrogen bonding. Enzymes align nucleotide by phosphodiester bonds to form a continuous strand. DNA replication. What first of all, what is need of replication? DNA carries all of the genetic information for life. One enormously long DNA molecules form haploid set of the chromosome of an organism, 23 of them in a human. The fundamental living unit is the single cell. A cell gives rise to many more cells through serial repetitions of the process known as cell division. Before each division, new copies must be made of each of the many molecules that form of the cell, including the duplication of all DNA molecules so that it can be equally distributed between two daughter cells. And what DNA replicates during cell cycle? In prokaryotes, DNA replication occurs during the stage of the cell cycle, while in eukaryotes it occurs during S phase of the cell cycle. What are similarities in replication of prokaryotes and eukaryotes? Basic mechanism of DNA replication are similar across organisms from prokaryote to multicellular eukaryotes. Origin of replication as usually contains 80 read sequence and replication is bidirectional from replication origin. Then replication is semi-conservative process. Replication occurs in 5' to 3' direction and is template-dependent process. Replication is semi-discontinuous process. DNA polymerase cannot initiate replication. It needs free 3OH group of extension of strand. DNA replication, complementary base pairing produce semi-conservative replication. Double helix unwind, each strand act as template. Complementary base pairing ensure that T single addition of A on new strand and G single addition of C, two daughter helices produced after replication. DNA replication and overview. Original double helix stand separate, complementary bases align, opposite template, enzyme link sugar phosphate elements of aligned nucleotide it into a continuous new strand. Experimental proof of semi-conservative replication, three possible model. First, semi-conservative, conservative and dispersive. Semi-conservative replication in this model, the two strands of DNA unwind from each other and each act as a template for synthesis of a new complementary strand. This results in two DNA molecules with one original strand and one new strand. This is the correct mode of DNA replication in biological organism. Second, conservative replication. In this model, DNA replication results in one molecule that consists of both original DNA strands and another molecule that consists of two new strands. This mode does not exist in biological organisms. Third, dispersive replication. In this dispersive model, DNA replication results in two DNA molecules that are mixture or hybrids of parental and daughter DNA. In this model, each individual strand is a patchwork of original and new strand. This model also does not exist in biological organisms. Next, this is Replicon's process. Michelson settle experiment confirm semi-conservative replication. How we can say that mode of DNA replication is semi-conservative? In semi-conservative mode of DNA replication, two parental strands unwind are used for synthesis of new strand following the rule of complementary base pairing. The semi-conservative mechanism of DNA replication can be best explained by a classic experiment conducted by M. Michelson and W. F. Suttle, outlined in Figure. Their experiment significantly explains difference between the semi-conservative, conservative, and dispersive modes of replication. First, Michelson and Settle labeled DNA in bacterium by flowing cells in medium containing either nitrogen 14 or the nitrogen heavier isotopes nitrogen 15. It has an additional neutron than the naturally occurring nitrogen 14 but not radioactive. Furthermore, they isolated pure DNA from these organisms and subjected it to CSCL density gradient centrifugation, leading separation of light nitrogen 14 and heavy nitrogen 15 form of DNA to different location is the centrifuge tube. To determine density of the stand, a technique unknown as density gradient centrifugation was used. DNA banded after equilibrium density gradient centrifugation at a position which matched the density of the DNA. Metal settle experiments confirm semi-conservative replication when bacterium grow in nitrogen 15 we are transferred to normal nitrogen 14 containing medium, the newly synthesized DNA strand and the nitrogen 14 while the parental strand had nitrogen 15. They checked the composition of the resulting DNA molecules by density gradient centrifugation found an intermediate band indicating a hybrid molecules containing both nitrogen 14 and nitrogen 15 DNA. This is the Mason subtle experiment. The mechanism of DNA replication tightly controlled process occurs at a specific time during the cell cycle, requires a set of protein and enzymes, and requires energy in the form of ATP. Two basic steps: initiation and elongation. Two basic component: template and primer. The mechanism of DNA replication. DNA polymerase, the enzymes that extend the primers, polymerase third, produce new strand of complementary DNA. Polymerase first fills in gaps between newly synthesized Okazaki segments. Addition enzyme proteins. DNA helicase unwind double helix, single stranded binding protein, keep helix open. Primase creates RNA primer to initiate synthesis. Ligase welds together Okazaki fragments. Mechanism of DNA replication. Figure. Origin of replication. Replication proceeds in both directions bidirectionally. From a single origin of replication on the prokaryotic circular chromosomes, replication proceeds in both directions bidirectionally from hundreds of thousands of origins of replication on each of the linear eukaryotic chromosomes. Origin of replications. Bacteria have one origin of replication per one chromosome. They only have one chromosome, one origin. What direction of DNA synthesis? The new strand of DNA is synthesized from 5' prime to 3' prime during semi-conservative replication. New nucleotides are added one at a time. The 3' prime hydroxyl end of the DNA chains forming new phosphodiester bond. The 3' prime to 5' prime parental strand act as template. Eukaryotic origins of replication. Replication initiation. DNA origin of replication, initiator protein bind recruits DNA helicase, opening of DNA strand, replication, initiation, primase and the RNA primers, replication elongation, DNA polymerase third must have 3' to add 2. Replication is finished. DNA polymerase first removes primers, fills gap using 3' ends. DNA ligase connect fracs uses 5' end. Replication fork. This figure represents replication fork. What really happens? DNA polymerase works as a dimer. Lagging strand must loop around to accommodate dimerization. Replication termination. The end of chromosomes cannot be replicated on the lagging strand because there is no primer available. Telomerase enzyme that contains RNA primase which extend the end of the chromosomes expressed insignificant. Telomerase form is thought of single-stranded cap around the chromosome end to protect them from being degraded. Chromosomes in are progressively sorted with each round of replication. Old cells with sorted telomerase undergo apoptosis, protective for animals, kills the old and possibly mutated. Telomerase is overexpressed in cancer cell. Hypothesis is that cancer cells do not undergo apoptosis because their telomerase do not shorten over time. While Why on lagging strand DNA replication is discontinuous? The lagging strand is the strand of new DNA whose direction of synthesis is opposite to the direction of the growing replication fork. On the lagging strand, DNA replication does not occur as a continuous stage, but it takes place in the form of short segment. Replication, the end of chromosome, telomerase adds an RNA primer complementary to telomerase sequence. Chromosomal replication proceeds by adding to the 3' end of the primer, fills the gap left behind by replication. Telomerase enzyme can also add DNA base pair to the template DNA. Complementary to the DNA primer are base pairs, using an RNA template to make DNA telomerase function as a reverse transcriptase called TERT. TERT is the telomerase reserve transcriptase. This goes against the central dogma, erroneously thought to be derived from a retrovirus. Synthesis of telomere DNA by telomerase. Replication at the chromosome level. Replication is bidirectional for circular DNA. The unwind at the replication fork cause supercoiling DNA topoisomerase enzyme that help relax the DNA by nicking the strand, releasing the twists, then rejoining the DNA ends. Example is DNA gyrase assembling newly replicated DNA into nucleosomes. The assembly of nucleosome after replication. And I have a question. How DNA duplex unwind at replication 4? Unwinding our separation is done by a specialized enzyme called as helicase. In prokaryotes as well as in eukaryotes, DNA helicase are the enzyme that use ATP as energy to separate the DNA duplex in the direction of replication 4. They progressively catalyze the transition from double-stranded DNA to single-stranded DNA. The role of helicase in E. coli is done by DNAB, while in eukaryote it is the performed by MCM227 proteins. And next question, what are the total what are total number of DNA polymerase? All the known DNA polymerase can add a nucleotide in 5' to 3' direction. They add nucleotide it at 3' OH of the growing strand of DNA. There are 5 DNA polymerase in bacteria as compared to the 14 DNA polymerase in eukaryotes and perform the similar overall function as the eukaryotic DNA polymerase. Uh, and Next question, what is the clamp loader? Clamp loader are the special class of protein complex that open up the clamp and also place it on the DNA and gamma, delta dash subunit of the DNA polymerase constitute the clamp loader in prokaryotes, while in eukaryotes the protein replication factor RFC is clamp loader. The RFC consists of 5 different protein subunits called RFC15. Clamp loader recruits sliding clamps at the primer DNA junction at the double-stranded stretch near the 3' end, which is going to be elongated. The sliding clamp in turn recruits the replicative polymerase at the time of polymerase switching bind of DNA. With the clamp complex is the following by ATP hydrolysis and subsequent release of the sliding clamp loader.